Hello everyone, I'm Sally Kochmar, Lab Manager for the Magee Women's Hospital Med Genetics and Genomics Laboratories, and I'm also the Education Director for AGT. It's my pleasure to welcome you to AGT's webinar series. For those of you who are not currently AGT members, please consider joining the organization so that we can continue to bring webinars like these to you, and we can be the voice of the laboratory technologist in the lab community. We currently have sent out all of the reminders for joining. If you haven't done that, if you were a member and need to renew, please do that as soon as you can. Also, we get a significant discount for the upcoming annual meeting, June 15th to 17th, which will be in St. Louis, and we have an exciting program lined up with a lot more speakers and platform presentations that I think everyone will really enjoy. So quick housekeeping notes before we go into today's presentation. Please feel free to ask questions during the session. You'll notice in the upper right-hand side of your computer screen there's a control panel, and in the lower portion of that panel you can type in a question or a comment and submit it to the webinar organizers. And you can do that at any time during the presentation. We'll reserve about 10 minutes at the end of the presentation in which I'll present the questions to Dr. Elsey. If you are participating as a group, please designate one person in the group to complete the online evaluation. In order for everyone to receive their certificates of attendance, that person will need to enter the names and email addresses of each person who is participating at your site today into the evaluation. As a backup measure, you can send in a sign-up form and fax it to AGT as well. Certificates will be emailed to participants approximately 30 days following the webinar. Today, Dr. Sarah Elsey will discuss clinical metabolomics for diagnosis of rare disease. Dr. Sarah Elsey is an Associate Professor of Molecular and Human Genetics at Baylor College of Medicine and Senior Division Director of Biochemical Genetics at Baylor Genetics. Dr. Elsey received a BS in Chemistry with a minor in Biology from Missouri State University and a PhD in Biochemistry from Vanderbilt University. She completed postdoctoral fellowships in Molecular and Biochemical Genetics at the Baylor College of Medicine and is a board-certified geneticist through the American Board of Medical Genetics and Genomics. She held faculty appointments at Michigan State University and Medical College of Virginia at Virginia Commonwealth University prior to returning to Baylor College of Medicine. Her research is focused on the discovery, pathomechanisms, diagnosis, and treatment of rare disease, particularly neurodevelopmental disorders. She's a member of several professional societies and has authored more than 80 scientific and lay articles. Dr. Elsey?
Thank you. Welcome, everyone. Today I'd like to tell you about some of the work that we've been doing in clinical metabolomics, as well as a historical perspective in the development of biochemical genetic testing over the past several years. The objectives for today's talk are listed here, and this is to include a review of inborn errors of metabolism and some historical perspectives, to describe untargeted metabolomics and how we are using it in clinical diagnosis, as well as the approach to using pathway studies obtained from, with the information obtained from the metabolomics approach, and how we can integrate this information appropriate to a specific patient. And then to give you some clinical case examples of how we utilize these approaches toward diagnosis. Metabolism and the genetics of metabolism have been studied for many years. More than 100 years ago, Sir Archibald Garrett first suggested that there were chemical individualities linked to disease and to predisposition to disease, and that these individual phenotypes and individual differences in biochemistry were perhaps genetic. He's considered the father of biochemical genetics. First made discoveries in the late 1800s with regard to hematoporphins in urine, and then later described papers on alkaptonuria and first proposed the idea of chemical individuality, and then proposed the concept of inborn errors of metabolism in 1909, which included albinism, alkaptonuria, cystinuria, and pentosuria, and first made the association between these congenital variations and Mendelian inheritance. This led us to a seminal paper in 1909 called Inborn Errors of Metabolism, and this really was the beginning of the work that really led to newborn screening, which many of us are familiar with today. And so inborn errors of metabolism really reside from, obviously, errors in our normal metabolism. And in a simplistic view, illustrated in this figure, shows a gene product, typically an enzyme or a protein, that is involved in a biochemical reaction where a substrate is converted to a product. And that would give us a normal metabolic output. However, in a situation where there has been a mutation or another DNA variant that results in a change in either the function of the enzyme or in the quantity of the enzyme, then prevents that substrate to product reaction from occurring. And in that process, we get disease. And so by comparing the normal to the disease phenotypes, we're able to develop diagnostic approaches to identify individuals who might be affected by these disorders. There are some clinical symptoms that are typically involved in and consistent across individuals who have inborn errors of metabolism. These can include metabolic acidosis, hyperammonemia, hypoglycemia or ketosis, and a variety of neurological abnormalities that include hypotonia or dystonia, seizures, lethargy, and developmental and cognitive delays. Cardiomyopathy may also be present in many disorders, as well as other myopathies and exercise intolerance. There may also be a family history of sudden deaths or other apparently life-threatening events. Organomegaly may be common in certain types of disorders, as well as progressive cognitive impairments, and there may also be some dysmorphologies, but these are more rare. Newborn screening results may be positive for a subset of disorders, and so early diagnoses are the result of a positive newborn screen. However, other individuals may be diagnosed later in life, and so it's always important to have a good family history. So over the years, we've developed many different biomarkers for diagnoses, and this may include accumulation of substrates or intermediates, the absence of or the reduction of the products of those biochemical reactions. There may also be accumulation of intermediates in a different or an alternative pathway because of the pathway blocks that have occurred. Most of these alterations are typically substantial and are easily identified, but many of them may be subtle. And so you may only see an abnormality when the body is physiologically stressed, either by starvation or by stress itself, viral infections, or other medical issues. In the diagnostic process, it's always important to have the right sample type. And for many disorders, plasma is utilized, and that would be the most common. It's a very clean substrate, much easier to obtain and analyze. Cerebrospinal fluid is often used as well, particularly for neurotransmitters, but that does require an invasive procedure. Urine, of course, is the least invasive body fluid to use. However, it is not always the most ideal substrate for finding, to identify biomarkers. It really depends on what the disorder is and if the analytes will accumulate in the urine. Urine also tends to be dirty and is a different concentration in every sample that's obtained, and so that adds to the difficulty in its analysis. Red blood cells, white blood cells, skin fibroblasts, and liver biopsies, these are also all utilized typically for enzyme assays. There may be difficulties in some of these analyses due to transfusions, and they often also require invasive techniques, particularly for the liver or skin analyses. So there's been a significant evolution of testing for diagnostics and diagnostic testing for IEMs. Early on, many assays were colorimetric. Enzymology was utilized, and it's still utilized today. There were bacterial inhibition assays, TLC was often utilized as well. We still see it in some instances, but it is much less commonly used, as are fluorometric assays. In more recent years, over the last 20 years, we've seen a greater use of HPLC, as well as GC and mass spec, and in particular tandem mass spectrometry. DNA testing is the most recent addition to our testing for diagnostic IEMs, and this may include either targeted gene testing or whole exome sequencing. And so screening for inborn errors of metabolism really has been done in the past for using urine most commonly. But this has evolved over the years, and now we routinely evaluate patients by evaluating amino acids, typically in the plasma, urine organic acids, and acylcarnitines in the plasma as well. Enzyme testing may be done in targeted situations, but those are not typically done from a screening protocol. Most individuals who are first evaluated for metabolic disorders have plasma amino acids, urine organic acids, and plasma acylcarnitines. And so the identification of an individual with an IEM can be based on clinical presentation, but again, sometimes that is very difficult. And so this first tier of metabolic screening tests, which I mentioned, including amino acids, organic acids, and acylcarnitines, is the typical and standard approach. This may be followed by some specific targeted testing if there's a specific group of disorders based on phenotype or family history. And the specific analytes that are identified then allow us to target in and focus on a specific metabolic pathway. This may be then followed by either confirmatory protein function studies like enzyme activity studies, or these DNA sequencing for the specific gene involved may be appropriate. As we try to expand and think more about our analysis of inborn errors of metabolism and the ability to diagnose even more disorders than we currently do, we are often challenged with the differences that we see in genetic variants and the interaction with the environment. And this is very challenging. Now, the environment can be the food that we eat, the stress that we place on our bodies, the exercise, can be circadian differences. There are many different environmental factors that influence our metabolism, and then the genetic variants that each of us carries, how those interact and influence interact with the environment and influence the metabolic output are going to be different across individuals. And so understanding these differences and how we might best identify the pathogenic changes that have occurred in the DNA is often challenging. So, of course, there are going to be intermediate phenotypes that we might be able to evaluate, including mRNA and protein, but also the metabolites. And it's coordinating and understanding this association between these intermediate phenotypes as well and the disease phenotype. And how can we best identify those markers and understand how they are contributing to disease, or maybe at least representative of disease, of a disease state. And so we've taken many different approaches over the years toward diagnosis of rare disease, including global and untargeted approaches that include chromosome microarrays, karyotyping, and whole exome sequencing, as well as specific and targeted approaches that might include targeted gene mutation analysis, and as well as biochemical screening, and including HPLC methods, as I've mentioned, and GCMS, as well as tandem assays, which would include newborn screening, which is limited to a subset of disorders in each state, acylcarnitines, and a variety of other targeted analyte tests that are typically available as single analytes or as panels of analytes in different laboratories. And so we feel that there really are some diagnostic needs out there to advance and improve our ability to diagnose the challenging cases. And we find that there are some significant challenges toward diagnosing patients with what we would consider undifferentiated phenotypes. Those include the phenotypes that fall into a very broad category that we commonly see in a genetics clinic. And this includes intellectual disability, developmental delays, seizures, failure to thrive, and autism spectrum disorder. There's a lack of clinical specificity in these phenotypes, which makes it even more challenging. Often many tests are needed to rule out metabolic disorders, and so this can require many different fluid types to have a full evaluation. It may also require multiple rounds of testing and multiple visits to a clinic, which adds expense and stress for every family. And of course, these costs of these tests may be very prohibitive. Some tests as well may be difficult to obtain, and there may not be an easy route or a satisfactory route really to tier the testing process. And unfortunately, many disorders do not have clinically available biochemical testing, so this is often challenging. Some biochemical tests, of course, may only be available by invasive methods, like liver biopsy or cerebrospinal fluid. And again, the pathogenic nature of the variants of uncertain significance that are identified in whole exome and targeted gene sequencing, these may be difficult to define and put into the context of disease if we do not have a biochemical test for confirmation of pathogenicity of these variants. And importantly, targeted tests are very specific, and they require that you know what you're looking for. And so this is important. And again, in these cases, we don't always know what we're looking for. These are undifferentiated phenotypes, which are challenging to diagnose. And so I'd like to think a little bit more about the methodologies and how we're approaching diagnosis of rare disease. Currently, most of the testing that we do is hypothesis-driven. We have targeted metabolic screening, targeted genetic screening in most situations, and which may involve specific analyte testing and enzyme screening, as I mentioned, as well as targeted gene testing. However, this approach is really changing. It's a significant paradigm shift in the approach to diagnosis. Rather than having a hypothesis-driven approach as the previous and really current in many cases, our current situation, we're really moving toward a data-driven and a hypothesis-generating approach, such that we approach from an untargeted means, considering, say, whole exome sequencing, and then narrow down and target that confirmation testing to confirm the diagnosis. This is really important because it ties together clinical assessments, omics technologies, and then also brings in targeted chemistry and biology and molecular biology, and brings all of these methodologies together in the end toward a specific diagnosis. And so when we want to think more about combinatorial functional omics, and we think about genomic methods, transcriptomics, proteomics, metabolomics, and phenomics too, right? And so all of these contribute together, contributing to the expression and function involved in the manifestations of rare disease. And we utilize readily genome or molecular data and of course phenome data from the clinic as well. And we know that while transcriptome and proteome data may be appropriate in the future, it's not quite ready for the clinic. Okay, and so we're not quite there yet. But the metabolome, and whether targeted analytes and then looking more broadly at all analytes that might be available is a reasonable approach and could easily contribute to interpreting some variants identified in the genome and their association with the clinical findings in the patient. And so by putting these together, the genome, the metabolome, and the phenome, we hope to get toward improved diagnosis of disease. So what is the metabolome? Well, the metabolome is the complete set of the metabolites found within a biological sample. Now, this sample can be a cell or an organelle or an entire organ or a biofluid. Okay, it's whatever that biological sample is defined, and then the approach of what kinds of molecules you identify are somewhat dependent upon how the tissue or the biological sample is treated. And so you may identify small molecules, lipids, or larger molecules. Our focus in the work that we have been doing is focused primarily on small molecules. Okay, so these are molecules that are smaller than 1,500 Daltons. These do include many lipids, but it is not a comprehensive lipid approach. And importantly, the metabolome is influenced heavily by not only the genome, which results in the proteome and of course can impact the metabolome, but there is a significant environmental influence as well as a physiological influence. And all of these entities work together to give us our metabolic output. And we do have a rationale behind thinking about a metabolomics approach. We really see it as the output of the DNA sequence. And so from the DNA sequence, of course, we ultimately get protein, and the protein generates, protein reactions generate small molecules. And we feel that those will give us some mechanistic insight into the phenotype, and really think of this as a bridge between the genome and the phenotype. Now, there are many different methods that are utilized with regard to metabolomics. Of course, there's an extraction method to target the certain kinds of molecules that you wish to identify. And then those extracts can then be introduced into either tandem mass spec, LC mass spec, GC mass spec, or NMR. And any of those approaches may work toward identifying rare or unknown molecule, known and unknown molecules in that particular biological sample. This may be a targeted or untargeted approach, but typical metabolomics that is for a screening method is going to be an untargeted approach. And again, it may be limited by molecular weight and also the molecule class. And so the approach that we're utilizing starts with biochemical extraction to pull out those molecules that are 50 to 1,500 Daltons. This extract is then run on 4 different UPLC mass spec platforms. Those ion features are captured and data are analyzed, and a library search based on retention time and mass spec signals are utilized, and molecules are identified. Then there's a QA/QC that's followed, and ultimately statistical analysis results in an output of z-scores for each molecule relative to a control population. These z-scores are then interpreted and within the context of metabolic pathways, which then allows us to identify abnormalities within those pathways and perhaps identify new biomarkers as well. These metabolites are then placed within the biochemical pathways as shown in this illustration from Cytoscape. Each of the molecules we identify is represented by a circle, and those molecules are all connected to a specific biochemical pathway. The metabolites that we're able to identify cover many different pathways, as illustrated here, from branched-chain amino acid metabolism to fatty acid metabolism, and purine and pyrimidine analyses, as well as TCA cycle and urea cycle and others. This allows us to get a very comprehensive view of the metabolic output within a particular biological sample. So a normal result that we might obtain from this analysis is illustrated here. The circles that you see are represented, are indicated on the left. Red is considered a high value, blue is considered a low value, grays and blacks are either within the normal range, or there may be a molecule that was not detected in the assay. And there are some molecules that are not yet in our library. The size of the circle is relative to the z-score. And so the z-score are standard deviations from the mean of the normal population. Illustrated in this image is the metabolic output of an individual with PKU. This is from a single individual, and you can compare this to the previous slide with regard to a normal output. You can see that an individual with PKU, and this is actually a patient who is on diet and considered well controlled, that they have many still abnormal metabolites, and that their metabolism is still quite not normal, even though we consider this a patient who's doing well. And if we zone in on just the phenylalanine metabolism pathway, you can see that that these metabolites are substantially abnormal, and that when compared to the normal population, an individual with PKU still has, even though well treated, still has significantly abnormal values. When we first developed this process, we did an extensive validation process, and we are still undergoing validation with new disorders all the time. This illustrated in this figure is a snapshot of some of the disorders that we evaluated. You can see the disorders are listed on the left with molecules related to that disease in the middle, and then on the right are the Z-score findings for individuals either who are affected by that disorder or not affected by that disorder. Affected individuals are illustrated in red, unaffected individuals are illustrated in blue. And you can see that if you look at PKU at the top, that individuals with PKU are clearly differentiated from the unaffected population. And as well, if you look down at many of these disorders, you'll see that these disorders are very well distinguished from the normal population. Importantly, all of the individuals on whose samples these were taken, from whom these were taken, all of these patients are on diet and on appropriate therapy. So these are not the original diagnostic samples. And so we might expect there to be less distinguishable characteristics. However, what we found is that most of these are quite distinguishable from the normal population. Down at the bottom, you'll see that there's a condition called thymidine phosphorylase deficiency. We have 3 different markers related to that disorder, including 2-deoxyuridine and 5,6-dihydrothymine, and those molecules do overlap somewhat with the normal range. However, thymidine was identified only in the 2 individuals who have the disorder, so that allowed us to have a distinguishing marker that confirmed the diagnosis. Illustrated in this image, it really is the power of a metabolomics process. This is branched chain amino acid metabolism, and I've highlighted a few different disorders in this pathway, and I'd like to focus on MSUD, which is illustrated in green. What we have in this pathway are different points in the pathway. Each number represents a molecule, and each diamond represents an enzyme with a particular disorder associated with it. MSUD impacts all 3 branched chain amino acids, including valine, isoleucine, and leucine, as shown in this figure. And when you're deficient in branched chain keto acid dehydrogenase, with that deficiency, you're going to see elevations above that enzyme block, which are illustrated in green, and you're going to see deficiencies below that enzyme block, as illustrated in green as well. In each of the graphs, what you see are individuals who are affected by those disorders. And so the individuals with MSUD are shown in green. Individuals with other disorders are shown in their appropriate colors based on the key at the bottom of the slide. And so what we find is that if we look closely at the MSUD cases, you see these elevations and reductions appropriate to the enzyme block. And this allows us to explore many of the molecules within the pathway, not just a single marker. If you look as well at propionic acidemia, down at the bottom in red, you'll see that individuals with that disorder have multiple elevations, and some of those elevations are very similar to cobalamin synthesis and methylmalonyl mutase deficiencies, illustrated in blue and yellow. However, individuals who have propionic acidemia are deficient in 2-methylmalonyl carnitine because of this enzyme block, which is illustrated in red. And so, again, the metabolomics process allows us to look at multiple molecules throughout the pathway, which allows us to look at the impact on the whole pathway and narrow in more closely on the diagnosis. And so now I'd like to spend the rest of the time talking about some case studies to illustrate the power of this process. And again, these studies are all going to use this combinatorial functional omics approach, where we have genome data, metabolomic data, and phenome data to pull together toward the diagnosis of these disorders. In the first case, this child presented at 19 months of age with global developmental delay, hypotonia, abnormal movements, an abnormal MRI, and oculomotor apraxia, as well as some other minor features, including facial hemangioma and constipation. He had a prior normal workup for everything that had been done. This includes microarray, a general metabolic workup that included amino acids, organic acids, as well as a lumbar punch for CSF to do amino acids and a neurotransmitter profile. He also had lactic acid and ammonia completed as well. So because of these prior normal workups and still no diagnosis, whole exome sequencing and metabolomics were ordered for this patient. Whole exome sequencing identified a number of different variants. One single heterozygous pathogenic variant in a gene called UROC1, which is associated with urocanase deficiency. Two heterozygous VUSs in a gene called ABAT, which is associated with GABA transaminase deficiency. He also had several single heterozygous VUSs in many genes, and most of these were recessive disorders, and they were not known pathogenic variants. Because of that, we decided to focus on the two primary variants identified in UROC1 and in ABAT. In UROC1, this was a single pathogenic variant because this was a frameshift mutation, so it's a pathogenic variant. And the question is always the situation where is there possibly an undetected pathogenic variant still present, which might lead to disease. So that's always a question in these kinds of cases. And then the second set of variants that we're going to explore include ABAT. And again, this is associated with GABA transaminase deficiency. These were two missense variants, one inherited from each parent. These were predicted to be deleterious, but unclear if they were or not because they had not been identified previously. Okay, so we're going to focus first on the UROC1 variant. Again, this is just a single variant. The father carries this same variant, but the mother is negative. And so we went to the metabolomics data, and we looked very carefully at the histidine pathway. Urocanase functions in the UROC1 pathway, okay? I'm sorry, urocanase is UROC1, and it functions in this production, in the conversion of urocanic acid to imidazolone propionic acid. If this is blocked, then what we see are elevations of imidazole propionic acid, urocanic acid, and a decrease in imidazolone propionic acid. Okay, so we would expect those in this deficiency. And we have seen other cases of deficiency for this gene, so we do know what we would expect and what we should see. However, in this situation, we found no alterations in this pathway. These molecules were all normal for the sample that we received on this child, and so this would suggest to us that a second pathogenic variant was not likely present. And so the diagnosis was likely not urocanase deficiency. And so we then went to the next set of variants and a completely different pathway, ABAT, and again looking at two heterozygous missense variants that both were predicted to be deleterious. And so if we now look at the GABA transaminase pathway, GABA transaminase functions to convert GABA to succinic semialdehyde. When it is absent and not functioning properly, we're going to see an increase in GABA. However, GABA can be very difficult to detect. It's very labile, and while the assay itself is simple to detect GABA levels, because GABA is labile, it's not, these analyses are not very reliable. And so we then went to look at the metabolomics data for this pathway, and we identified a molecule called 2-pyrrolidinone. Prior to this analysis, we were not aware of 2-pyrrolidinone and really thought of it only as a molecule associated with drug metabolism. However, what we found is that when GABA is increased, it is converted to 2-pyrrolidinone. 2-pyrrolidinone is a lactam cyclized version of GABA, and And so now, based on this, we've been able to identify 2-pyrrolidinone as a biomarker for ABAT deficiency. And we can identify this in plasma, which is very important. And so to illustrate this biomarker identification, what we found is that if we have GABA, that GABA can be converted to 2-pyrrolidinone, and this is a normal process. This occurs in all of us. However, when GABA is significantly elevated, what happens is we get a significant elevation of 2-pyrrolidinone because you get a greater conversion of GABA to the 2-pyrrolidinone molecule. And so we have 2 independent samples from this child, and you can see that we have a Z-score that is well above 6. This is a very, very high Z-score. And we have our normal unaffected individual ranges of Z-scores. And so this elevation is consistent with elevations of GABA in the plasma. In this process, we also identified several different samples that had elevations of this molecule, but these individuals did not have mutations in this gene. However, the patients were taken by gabatrin or topiramate, which are anti-epileptic drugs that are known to increase levels of GABA, which then will increase the level of 2-pyrrolidinone. And so these are drug-associated metabolites that we've identified here. And we know now that these drug-associated metabolites can impact and maybe detract from our knowledge and with regard to approaching a correct diagnosis for these patients. And so we're now quite aware of the levels that we would expect to obtain with vigabatrin or topiramate treatment, as well as other medications, and know that they may be confounders with regard to the proper diagnosis of this disorder. And so GABA transaminase deficiency is a rare disorder. There have only been 3 other families reported. A common phenotype across this disorder includes psychomotor retardation, hypotonia, hyperreflexia, lethargy, and refractory seizures and EEG abnormalities. There typically also is early death associated with this disorder. Diagnosis has involved in the past CSF neurotransmitter analysis to look specifically for GABA. Because GABA is not a stable molecule, as I mentioned, specific testing, collection tubes, and handling must be requested in order for GABA to be evaluated. It will not be evaluated as a normal process, as a normal neurotransmitter identified in these analyses. It must be requested separately. And so, to summarize this particular case, what we found is an individual with a UROC1 mutation. However, metabolomics was normal for urocanic acid, and an individual who had ABAT mutations, which suggested GABA transaminase deficiency. And we found that 2-pyrrolidinone elevation in plasma metabolomics helped us to confirm the diagnosis in this individual as GABA transaminase deficiency. And so this was important because we were able to not only diagnose the disorder, but we were also able to diagnose it in plasma, which going forward means that we don't have to require cerebrospinal fluid, which is really wonderful for these patients and families. Okay, the second study I'd like to present is a child that presented initially at 11 months and then was lost to follow-up until about 4 years of age. The child has hypotonia, severe developmental delay, and occasional periods of stiffening that were much like seizures. He was initially diagnosed with cerebral palsy. His exam at 4 years of age showed basically normal growth, but he was quite underweight and static cognitive development at 3 to 6 months of age. He had a hypotonic facies with mild dysmorphic features and a substantially decreased muscle bulk. He also had frequent oculogyrate crises. This child had an extensive workup, and you can see here the many different tests that this child had. That included CSF as well, which required spinal tap, as well as a muscle biopsy. And so you can see that he's had some invasive testing that's been performed. Whole exome sequencing analyses revealed mutations in aromatic amino acid decarboxylase. Aromatic amino acid decarboxylase deficiency is a known disorder. And however, these were variants of uncertain significance, and they were, while they were predicted to be deleterious and they were confirmed to be inherited from mother and father. It was unclear if these were going to be diagnostic and causative of his disease. And so this disorder, as illustrated in the pathway here, functions in dopamine and serotonin metabolism. AADC is the enzyme indicated here, and when that is deficient, you don't get the conversion of DOPA to dopamine or 5-HTP to serotonin, and as a result you get an increase, you get a backup, and you get an increase in 3-methoxytyrosine, and then decreases of all the downstream products, including dopamine and serotonin, VMA, HVA, and 5-HIAA. Now, this child had a second neurotransmitter analysis. After the whole exome sequencing results resulted, he had a second lumbar punch for CSF neurotransmitters. And in that analysis, he had HVA that was undetectable, 5-HIAA was also undetectable, and 3-methoxytyrosine was significantly elevated. Okay, and so CSF is the usual body fluid for this diagnosis, and so this diagnosis was confirmed for this child, and that he had AADC deficiency. However, we obtained at the time that he had his CSF neurotransmitter profile, we also obtained a plasma sample, and we were able to run metabolomics on this and develop a plasma metabolomics profile, and that's illustrated here. And you can see that we identify nucleotides, lipids, carbohydrates, a variety of cofactors, and amino acids as well. And you can see that 3-methoxytyrosine is significantly elevated and is really the only molecule that has significant elevation in this patient sample. And so again, this is another illustration of the same molecule in this child as significantly elevated. And this is the first time we've been able to identify this molecule in plasma versus CSF, and so we were able to confirm the diagnosis by utilizing a plasma sample and not requiring CSF. We also found other abnormalities that we could identify using metabolomics, and again, which are illustrated in the graph on the right. And again, for this disorder, as I mentioned, you see this elevation of 3-methoxytyrosine, but in our metabolomics, we're also able to see the reduced dopamine and the reduced VMA. The patient's sample is illustrated in red in the graph on the left. And so you can see the significant elevation and reductions showing full pathway impact of this anomaly. Okay, and so the third and last case I'd like to mention is an interesting set of families, all with a similar diagnosis. The first family is illustrated here. Simple family, single child. Child presents at 2 years of age with global developmental delay, increased tone, normal growth, no seizures, and no significant history at all. Child had a pretty extensive workup, including the usual metabolic profile, brain MRI. He had CSF analyses, so he had a pretty standard but sufficient normal workup. And so metabolomics was ordered. And in this analysis, we very easily saw an elevated succinyladenosine. Okay, this was the only significant molecule that was really elevated in his entire profile. And that's illustrated here in a snapshot from our cytoscape pathway. And again, here illustrated in the graph on the left is his significantly elevated succinyladenosine compared to all the other patients who are unaffected by this disorder. We did targeted follow-up studies to confirm this diagnosis. We confirmed the levels of succinyladenosine in urine, as shown here, and you can see that they are substantially elevated. And we also took his previous CSF sample and also measured succinyladenosine in that sample. And so you can see that we see significant but consistent elevations of this molecule. And so the VUSs, so we then did targeted sequencing and identified 2 VUSs in the ADSL gene, as illustrated here. And so this confirmed that this child has adenylosuccinate lyase deficiency. Now, this disorder, there are fewer than 100 cases reported. It is a neurometabolic disorder with encephalopathy, developmental delay, intellectual disability, and motor delays, autistic behaviors, as well as seizures. So seizures are a key finding in this disorder. Diagnosis typically requires urine purine analysis that most of the time is centered on succinyl-adenosine. As illustrated in this pathway figure, ADSL functions in 2 points in this pathway, including here in the conversion of SICAR to ICAR and in the conversion of SAMP to AMP. Okay? And in the blockage of these pathways, what we find is that we get the succinyl-adenosine elevation as well as a SICAR elevation. SICAR is labile and is challenging to detect, so our analyses and others are focused on succinyl-adenosine. And so since we identified that first patient, we've identified 2 additional families, a total of 4 affected individuals with this disorder. All of our cases were confirmed with targeted testing and ADSL mutation analysis. And with this, we've been able to expand the phenotypic spectrum of the disease and realize, too, that the metabolomics approach allows us to screen for a variety of phenotypes in a single sample. The 3 samples that we use for metabolomics analysis are illustrated here. And I'd like to summarize these families. So the first family that I mentioned is family 1. These individuals had an atypical presentation with novel variants. So no seizures, developmental delay, but not a typical presentation for this disorder. And this patient had VUSs. In the second family that we identified, this individual had a known mutation and was homozygous for that known mutation and had a typical presentation. This individual had seizures, developmental delays, and had otherwise what we would consider a typical presentation. The third family is illustrated here, and in this family, the child that came to the clinic is illustrated here in the middle, and we have 3 boys with developmental delays. And however, the middle child had more significant phenotype with seizures and oculomotor apraxia, and so our testing focused on that child initially. And so while metabolomics and whole exome sequencing were ordered for this child, and while the whole exome sequencing was being completed, our metabolomics analysis identified elevated succinyl-adenosine in this child. We were then able to bring in the other children in the family and confirm their findings, and we found that the younger boy also has this disorder with elevated succinyl-adenosine. However, the older child does not have this condition. So 2 out of the 3 boys in this family are impacted by this condition. And so they have an unusual presentation of a known variant. They have the same variant that was identified in family 2, but their presentation is very different. And so this shows us even more of the phenotypic heterogeneity that we see across many of these disorders. This slide illustrates the targeted analyses that we then did to follow up and confirm the findings in these patients. And because we did the metabolomics on the first in the third family, because we did the metabolomics only on the middle child, we did not have to do metabolomics for patients 1 and 3. For the older and younger siblings, we could do targeted analyses, which are faster and much less expensive for the families. And so we were able to come to diagnoses very rapidly for this family. And so through this process, we've been able to confirm many new diagnoses in families. We've also been able to rule out diagnoses based on the metabolomics approach. And so these are just partial lists of where we are at this point, but we're adding to this list every week. And so we find that this approach is cost-effective supplement to newborn screening, and that we're able to identify patients at the mild end of the spectrum, and as well, importantly, confirm molecular analysis results, because variants of uncertain significance are a substantial issue for molecular testing at this point. And so complementing biochemical testing is a significant addition and positive approach toward reaching diagnoses. And so these studies have all been done with a number of different people. Some of those people are listed here, include clinicians who refer us interesting patients so that we're able to confirm the diagnoses, as well as the individuals who've been intimately involved in the data analysis. So thank you, and I'll be happy to take any questions.
Thank you, Dr. Elsey, for a very informative and interesting presentation. As a reminder, you can submit your questions to Dr. Elsey by typing in the control panel to the right side of your screen, and then we'll get the questions to her. Do you see metabolomics working hand in hand with pharmacogenomics in terms of being able to treat some of these patients?
Yes, actually I do. I think that's maybe two questions. I think that from a pharmacogenomics perspective, we are able to identify drug levels and relative drug levels in many samples that we receive. And so I think that in association with pharmacogenetics, that targeted or untargeted analyses with regard to drug medications, drugs and various other medications, I see certainly in the future. We're not there yet, but I think that that's coming. With regard to treatment, yes, I think that we've followed a number of patients from a treatment perspective for a variety of different conditions, and we know that not everybody responds to treatment in the same way, and so and that may be because they actually may have a second condition as well that's impacting their diagnosis and the treatment for that initial condition, or perhaps they have other variants that are contributing to their lack of response to the treatment. So we've been able to follow many patients in this way, and so I think there's great benefit. I think we're still at the beginning in that process, but I think that's where we're going.
Thank you. We do have a question. Someone says they have a patient who has developmental delay, hypotonia, seizures, and has had cytogenetics and microarray performed, and they're all normal. The child is only diagnosed with autism, 4 years old, and still not talking, and she has developmental delay, and her seizures are under control with meds. Would whole exon sequencing be recommended since all the other tests have come back normal?
Yes, I would recommend whole exome sequencing in combination with the metabolomics approach. I think that would be most beneficial. The symptoms you've described are the same kinds of symptoms and features that we see in many of the patients that we evaluate. And we find that we are able to diagnose about 10% of those individuals with a metabolic condition. However, the other 90% are going to have another kind of developmental disorder, and the phenotypes may not be distinguishable, in the end. So my recommendation would be both, either both done together or done in tandem.
Thank you. Another participant is asking, what is the significance of the double slash lines to the right of the male parent in your pedigree of family 3?
Oh, okay. Let's see. Let's go back and look. Oh, so in those cases, so he had there was a divorce basically. So so that mother had another child from another partner. So if you'll see the 26-year-old off to the far right is only related to her, is not related to her current husband. So I hope that's clear.
Thank you. We have a participant from Mexico who wants to know what can they do in their regular approach in countries like Mexico where the newborn screening is basically for hypothyroidism. What do you recommend?
Yeah, it's challenging. So certainly, newborn screening that will identify individuals who are impacted by somewhere around 30 to 40 disorders, depending on the particular screen. Would certainly recommend that. I know it's not readily available in Mexico, and that you're going to have to send that sample out to another laboratory, likely in the U.S. or Canada or elsewhere. And metabolomics is going to be a much broader screen. Right now, I think from a newborn screening perspective, I would recommend targeting a newborn screen with a blood spot that can be where you can get results very quickly and inexpensively. Metabolomics is more expensive, so and does not give you results within a few days, whereas newborn screening at least is going to get those really critical disorders identified right away. If those are negative and there is concern for a metabolic condition, then I would recommend the metabolomics.
Great. Those are all the questions that we have at this time. So I'd like to thank everyone for participating in the webinar. And shortly, the webinar participants will receive an email from AGT that includes the link to an evaluation. And please take a few minutes to access it, provide your feedback on today's session. And a reminder that this webinar today is fully recorded and will be posted on the AGT website shortly. We usually have it up within a few days. And so those colleagues of yours who were not able to see the live presentation today, or if you would just like to go back and review Dr. Elsie's slides, they will be available on the AGT website. We hope you will join us for upcoming webinars over the next few months, and we'll send out notifications to members once registration is available for the next webinar, which we hope to have another one scheduled for February or March. And again, reminder that AGT's 42nd annual meeting will be in St. Louis, Missouri from June 15th to 17th, and that we're now accepting abstracts which are due by February 1st, so please get those abstracts in. Thank you very much. Thank you, Dr. Elsie.
Thank you.